This is REC. REC is also known as the alpha helical lobe of the double-lobed protein Cas9. The alpha helical lobe is where the CRISPR-RNA and tracrRNA duplex interacts with the protein domains of the Cas9 endonuclease. So this is the upper lobe, the alpha helical lobe of the Cas9 endonuclease. This is the NUC, which is the second lobe. NUC is the home to the protospacer adjacent motif, the HNH nuclease region, along with the RUVC nuclease domain. The PAM sequence, PAM sequence, is where DNA unwinding is initiated. It's also where the RNA-DNA heteroduplex is first formed. And the PAM region is going to be right here on the NUC. NUC domain, NUC lobe. HNH nuclease domain is a domain inside of the NUC lobe of Cas9. HNH cleaves the single-stranded DNA complementary to the guide RNA. So your guide RNA is going to come back up to the alpha helical lobe. RUVC nuclease domain, here, we'll do in a different blue, is also a domain inside of the NUC lobe of Cas9. RUVC cleaves the unwound DNA at the non-complementary strand. This is the guide RNA. It, in Steptococcus pyogenes, is 20 nucleotides long. This endonuclease will scan the chromatin of the cell and check for complementarity of the PAM sequence. When the PAM sequence complements, and with Cas9 the PAM sequence is nucleotide GG, it will pause on that DNA and check for complementarity of the guide RNA. If the guide RNA and PAM sequence are complementary, then the endonuclease regions HNH and also RUVC will digest the two phosphodiester bonds on the backbone of the DNA and create a double-stranded DNA break, cleavage. The lower stem the bulge, the upper stem, the nexus. And finally, the hairpin. While the endonuclease travels and scans the DNA of the host cell's chromatin, it pauses at each PAM sequence to check for complementarity of the guide RNA. If the PAM sequence is complementary and the guide RNA is complementary to the 5' strand of unwound DNA, then HNH and RUVC will cleave the phosphodiester bonds of the double-stranded DNA, which in turn initiates either non-homologous end joining or homology-directed repair, both of which create indel mutations via insertions and deletions. The final full protein is the endonuclease known as cas9 and is found naturally in Steptococcus pyogenes as an adaptive immune system which scans bacteriophage DNA in order to cleave pathogenic sequences. The guide RNA can be synthesized to include any 20 nucleotide long combination, making it an editable endonuclease. The endonuclease can be introduced to the ovum during germline editing to create the indel mutations to desired knockdown protein expression in differentiated cells. Just going to put my mark right there. Germline editing is very efficient. You affect the entire organism all at once inside the ovum. You CRISPR the ovum through either microinjection or electroporation, and you do so before the sperm is introduced. So before it becomes a zygote. This procedure affects the entire organism at once, making the ovum zygote stage the most efficient time to edit the genetic information. This is applicable in both Homo sapiens and chimeras. Somatic cell editing is more difficult because it is the genetic modification of a mature organism's microbiome. In somatic cell editing, the precursor cells have to be edited, and in order to introduce the modification, one must clone the RNA duplexes along with the specific guide RNAs into a selectable virus-producing plasmid. Also cloned into this plasmid contains the element cas9. This plasmid should, when introduced to the cell's ribosomes, produce viral particles coated or enveloped by specific proteins which will articulate with only the receptors of the precursor cells. This allows for cellular-specific targeted interactions among the editing virus and host cell. The host cell in this case is the precursor cell to the cell one wishes to edit. The process of inoculation allows the individual or the individual's microbiome to become partially genetically modified and is highly inefficient when compared to germline editing.